first thing I want to ask, what is structure prediction? Anybody want to throw some ideas out?
From an amino acid sequence, predict the 3D fold of protein.
Yep, I mean, that's pretty much exactly right. Given the linear amino acid sequence of the polypeptide that you're interested in, what is the 3D structure of that? So, like we said, we have a sequence, we have all the amino acids and their single letter abbreviations, and then we're asking what's the tertiary structure of that protein, or if there's multiple chains, what's the quaternary interaction, etc. So this is what we're interested in. This is the question we're asking. How do we teach a model to do this? Before we get to all that, what AlphaFold is, and etc., let's do a little bit of background. So we're going to talk a little bit about some biophysical principles, and the first one of which is Anfinsen's dogma. And the real idea behind this dogma is that the native structure of a protein is typically found at a free energy minimum. So we have this gigantic free energy landscape of proteins, highly flexible, highly dynamic proteins. But the native structure, according to Anfinsen, occupies a low energy state, or minimum in this energy landscape. But Levinthal introduced a question. He asked, how can proteins fold spontaneously or really, really fast, essentially, if they have so many degrees of freedom? For instance, 101 residue protein has over 3 to the 200 power different conformations. And that's an underestimate. That's literally just manipulating the torsional degrees of freedom of the backbone. So there's an incredibly vast landscape of protein structure. So how do we even fold proteins in nature? How does nature even do it? Well, I think the key insight is that the energy landscape is not a golf course. There's not a single hole that you fall into and you have a flat landscape besides that. No, that's not really how proteins work. That's not the energy landscape. It's more shaped like a funnel where there are directional cues of where you need to go, and there are certain structures within that can be shared between other structures. For example, alpha helices are a repeated secondary structure element that a lot of proteins adopt. And so you actually follow certain trajectories or the gradients, or even like start heading down this path even without knowing exactly where that minimum lands. And so this is the key insight into why proteins and how proteins can fold. So one of the most important things for structural biology is this CASP tournament, which is the critical assessment of protein structure prediction. This started back, I guess, 1994. It's a biannual competition that essentially they ask experimentalists to submit structures that have just been recently solved, unpublished, so they're not available to the wider public. And then they ask contestants to predict the structure of that protein given the amino acid sequence. So this is really a very rigorous but blind assessment of how well your models can actually predict the structure. Because around this time when the CASP started, people think, oh, structure prediction, we got this is easy. But then they started doing these CASP predictions, and they were like, whoa, we're actually not really good at all. So this GT is a measure of like global similarity between proteins. And as you can see, at the really difficult targets, only around 20% of the C alphas were aligned. So like, we're really not that amazing, at least back then. And as you can see, there kind of has been gradual progress over the years, gradually improving the prediction performance. There's a few notable things I want to mention. Around CASP 3, which is around 1998, the predominant method was really like these fragment-based assembly methods. So the idea here is, you know the amino acid sequence of the protein you're interested in. You can find fragments that have the matching sequence within the PDB, and we have structural elements corresponding to that fragment. You can just swap it out and add that fragment, and then you have that still a long polypeptide chain. You keep replacing fragments until you have like a folded structure. And there's some sort of score function, energy function to guide that process. But as you can imagine, that's kind of computationally expensive. You're like taking a sequence, you're aligning it to the PDB, grabbing that structural element, grafting it in, and then finding other pieces, and just it's really kind of a complicated process. And it also didn't work very well. So that's kind of where we started though. And then jump ahead a little bit. Gradually, yeah.
So how to define easy and difficult targets?
That is a good question. And the way that CASP defines it is if there's like a homologous protein already solved in the PDB. So if you have like a sequence that is very similar to a protein that has already been solved structure, that's probably an easy target because there's a homologous template you could use to at least get you started with that structure. These guys at the difficult end really have no structural or like sequence similarity to anything in the PDB. So they're probably a lot harder to actually predict their structure. Well, I have already kind of hinted at the fact that homology is a really useful thing for structure prediction. One way of doing that is looking at a bunch of similar protein sequences from a variety of different species. So you can form something called an MSA, or multiple sequence alignment. So here you take sequences that are all very similar, you stack them up at just rows and rows of sequences, and then you can look down the column. Once they've been aligned, you can look down the column and see how sequences are changing in the different species. Now, imagine you had a species that made a random deleterious mutation to a position, maybe in the core or something, and change it to, I don't know, something that's vastly different from what it originally was, something that's charged. That would typically disrupt that protein's function, kill the organism. That would not be good. So one way to compensate that would be if you have that residue mutated, and then you have a residue that interacts with that, you can make a compensatory mutation to the other residue to compensate and adjust for the changes in that first residue. So, for example, we had a hydrophobic residue in the core of a protein. We changed it to a positive charged arginine or something like that. We mutate a residue that was interacting with it to a negative charge. So now you've got a very favorable interaction, and that could probably rescue that protein's function and save that organism. So as you can imagine, if you look at a variety of different positions and you see these patterns of co-evolution of sequences or positions that are changing at the same time as other positions, maybe they're actually interacting. So that forms the basis of the thoughts of these co-evolutionary analyses methods, and that's also kind of the backbone of why all the structure prediction networks that we're seeing now, AlphaFold, Rosetta Fold, all these are able to work because of this co-evolutionary analysis, and they're all doing this, as we'll talk about later. This is also around the time that deep learning really started taking off, specifically for protein design. And originally around this time, they were kind of using these convolutional neural networks that like looked at a protein like it was an image, and then it would scan across that protein and try to recognize patterns within that protein in order to predict its structure. So it would form like this pairwise distance matrix or something like that, and then look at every pair's interaction with one another, and eventually try to come up with some sort of hypothesis of what that structure looks like. This is like around the era of TR Rosetta, if you've heard about it. The next major points, a few years later, CASP 13, this is where AlphaFold 1 was introduced. It was still a convolutional neural network that relied on MSAs, multiple sequences alignments, but it also raised the bar a lot. So, well, not at the easy end, because those still didn't really change much, because there's a lot of homology in the PDB, and so you can kind of base it off that. But for at least the hard targets, it raised the bar pretty good. And so this showed like a dramatic improvement over the previous years. It really showed the power of these deep learning-based methods for structure prediction. Now, it wasn't an end-to-end network. The model itself actually produced these predictions of distributions that would constrain what the shape looked like. And then they would use some energy-based minimization method, something like in Rosetta, and they would guide using these predicted distributions as potentials to guide it into the final structure. So it wasn't a fully end-to-end deep learning-based network like the new ones that we're seeing now, but it was really one of the first stepping stones. I'm going to stop talking about CASP here, and we'll talk about CASP 14 when we go into AlphaFold, because that's AlphaFold 2, sorry. And yeah, we'll leave you on the cliffhanger here to see what happens next. But we're seeing some nice improvements as we go throughout the years. Some structure-based prediction metrics that you'll likely hear. Probably the most common thing to do when you're looking at your structure is just to try to align them. And the most common metric you'll see is RMSD. So it's just really you take two proteins, align them, and then calculate the average distance between the atoms after alignment. Often you'll see RMSD C alpha or RMSD C alpha 95, which is just like they remove 5% of the outliers, which really accounts for like the floppiness of the termini. One of the problems with RMSD is it is length dependent. So as you grow a protein, there's this power law scaling with RMSD as well as some of these other ones that have length dependence. So it might not be the greatest metric when you're comparing proteins across different sizes, or kind of computing an average for a model or something like that, because there is this length dependence. It's in units of angstroms or some other length metric, and then it's just a positive value, with 0 being the minimum. Another useful one is local distance test, or LDAT. This is a measure of local similarity. So one question I have for you guys is, why would we want a measure of local similarity when we already have like RMSD? Like, this allows you to calculate the entire align two structures and then compute the error of that. Why do we want like a local alignment? Any ideas?
So a big picture view might hide like smaller details where this metric will pick up.
That's a very good point. Yeah, you're missing out on some of these atomic details that might be in the models. Anything else?
There may be some motions between the domains.
Domain motions?
Yeah, I was gonna say that if the domain is separated, it's gonna have a high RFD, but maybe domain B are like perfect copies of each other.
Exactly. Yeah, that's a good point. Yeah. Yeah, Joe?
If sections of the protein are like unstructured, you might just have like it might take the average down to the rest of the structure.
So those are all excellent points and exactly why we want local similarity checks as well. So these are able to measure like the local interaction of a very specific structural neighborhood around each residue, which is super useful when you're like looking at the domain structures of proteins. So if you have like a multi-domain protein, if it's supposed to look like this and it's actually like this, the domains could be folded perfectly right. But the actual interaction between the domains could be entirely wrong. And which would really screw up your RMSD calculation, which means, but that might give you a wrong impression that the entire protein is folded, or misfolded, sorry, when in reality the domains are actually folded properly. There's not really a unit for this, but it kind of ranges from 0 to 1, with 1 being a perfect alignment of local structures, and it's not length dependent, which is also really useful for comparing across sizes.
So that is a distance of 1 equal to RMSD of 0? I mean, 0.
Yes. The local distance. Well, yes. I mean, a local distance test of 1 means everything, the local environment is perfectly aligned.
So the big picture.
So the big picture is probably also going to be aligned. It might not be perfectly aligned because of the local distance test has like a thresholding, so like it only looks so far away. But if it's outside of that range, it's not really being counted in the local distance similarity check. I don't know if that made sense, but if it's a large protein, it might not be the exact same, but it should be wrong. Cool. Any questions? Okay. The next one is global distance test. This is another way of checking global similarity, but it's supposed to be more robust than RMSD. And I'll try to give you a few reasons why. The first one of which is that it looks at these two structures at multiple different alignments. So it aligns it in multiple different ways, which can account for different outliers that you might have in the RMSD calculation. It's more robust to like deviations in these really flexible loopy regions or the termini of the protein. So it's more robust in that sense. And yeah, so it's typically a percentage and ranges from 0 to 100, 100 being perfect global alignments. And it typically looks at only C alphas. But there are extensions to look at all atoms, including side chains. And the final one is this template modeling score, just another way of doing global similarity. The main benefit, I think, here is that it is not length dependent. So they have this normalization factor that really gets rid of this length dependency, which can make it really important. Make it really useful for comparing across different structures, especially if you're like missing residues or it's like not resolved or something like that. This TM score is pretty useful. I think this TM score also requires the sequence to be exactly the same. So if you have different sequences, then you can't really do that, but you can also do the other ones. Cool. Any questions?
So what TM score align? But correspond with RMSD score?
They should be like roughly the same. So like if you have RMSD at 0, you have a perfect TM score, you have a perfect global distance test, and you should have a roughly perfect local distance test. besides that, it's hard to exactly say because RMC is so length dependent.
Domain.
Yeah.
And what's the
Oh, sorry.
what did I'm sorry. What did the different like the primary difference between flow with distance test and honesty?
Sorry, what?
What is the actual difference between?
Yeah, so RMSD, you do a single alignment, and then you calculate the distance between every, the error in the atomic positions of every residue, or every atom, or if you want to like reduce it to see how close you can. But global distance test, you do multiple different alignments at different thresholding values. So you align like, you align it, and then you calculate what percentage of residues are aligned within, like have less than a threshold value of error. So you're aligning multiple times and you're thresholding it at different error values, and then you average that to compute your final global distance test. So yeah, it's slightly different, but it's a little bit more robust because of these multiple averages.
Thank you.
So with all the tools that we're learning this week, could you give me a bit of a application for when each of these metrics should be used?
Yeah. So I think the most interesting thing is that when we look at models like AlphaFold 2, they'll actually give you a predicted version of some of these metrics. Like it'll give you a P or predicted LBDT. It actually measures its own confidence in the local structure of every residue. It also will give you a PTM. So it also give you a rough estimate of how good the global structure is. So I think that's probably the most interesting thing. RMSD is just like probably the simplest thing you can do. It's usually pretty easy to do. Like PyMol, you can just really align these things. That's mostly what that does. It's slightly different algorithm, but it's mostly just RMSD. So like I think that's probably the most useful one. Global distance test is really just used by the CASP people. So yeah, I've never used that in practice.
And what are the threshold values for similarity for each metric? Like, if I have a RMSD of 1, is it close enough or like?
That's a very interesting question, and it depends a lot. For RMSD, it's hard to say exactly what you want. It depends on what you're doing. So if you have like a catalytic active site, you want atomic detail. You want everything to be super precise. But if you're just trying to look for the overall global fold, the minor details don't really care. It doesn't really matter. So it really depends on the problem. But for at least some of these, like TM score, I do know some of the thresholds. So I think most of the time, if it's like 0.5, it's usually the same structure is usually the idea. Anything above 0.5 means like more homologous to the target, but less than 0.5 means maybe like not homologous. Yeah.
If you don't mind me adding to that, the EMBL-EDI website has a whole tutorial on describing those confidence scores and how they can be applied. And then the specific rough cutoffs you can have for poor quality and high quality.
Yeah, we'll talk about this later, but AlphaFold has a giant database of a bunch of predictive structures made with FAQ that has some like interpretability of like what do these values mean? Why are they useful? And we'll talk about that too a little bit later. Cool. Okay, why do we want to do structure prediction? Well, there are many different applications, one of which molecular replacement. So people are trying to solve all these structures, and it can be really time consuming, it can be difficult. They're trying to build atomic models into these crystal densities or the diffraction pattern, and then the biodensities that people are creating. You want to like pump the models out of there so that it matches your experimental data. So predictive structures have been used to fill in or enhance these structures and improve the resolution of some of them by fitting the density better. You can also use it to help interpret experimental results. So if you have some sort of interesting result that maybe these two proteins are interacting or something like that, these two residues are interacting, you can maybe validate some of those experimental results or corroborate those results with some predicted structures. It's also useful for functional testing and hypothesis testing because you can make a predicted structure. You can ask questions like, does it look like it coordinates at maybe a zinc ion or something like that, or maybe it has a very specific pocket shape, something like that. You can like start creating some hypotheses of what that protein may be doing functionally or what it may be interacting with at a higher systemic level. And then I think at least what I'm more interested in is the design or the engineering aspects. You can totally use this as a starting place for design, as we'll see later. We'll use a bunch of predictive structures as inputs for our models. And that's really what we're going to be doing in this bootcamp.